God bless mommy. God bless daddy. Stand up. Stand up. Stand up, girl. Stand up. Sit down. Studies. Attention. Studies. Attention. నిల్చోవాలి నిల్చోవాలి Studies. Attention. కూర్చొని చెప్తా Studies. Attention. జనగణ మన అధినాయక జయ హై భారత్ క్లాస్ వస్తాను కాదు జనగణ మన అధినాయక జయ హై భారత పంజాబ్ సింధ్ గుజరాత్ మరాఠా ద్రావిడ Lavalinga, India, Himachal, Yamuna, Ganga. Studies, attention. Studies, attention. This pass. This pass.
Bob, stand up.
Stand up. Sit down. Stand up. Studies. Attention. Studies. Attention. Studies. Attention. Studies. Attention. Studies. Studies. Attention. Studies. Attention. Studies. Attention.
Mummy.
Karu. Anna.
Daddy.
Anna Radi. Ini mami.
Daddy.
Mami.
Daddy. Dengar. Daddy.
Daddy.
చిన్న నారి చిన్న నారి
కాదన్న డాడీ.
నా చిన్న నారి
చిన్నదా ఇది? చిన్నదా ఇది? లే పోరడా అమ్మది తప్పు కప్పుడు వస్తుంది చిన్నడా. సారీ
నా మాకు నారి
అండి. కాలు పోగి చక్కరే వాళ్ళు పోసి చక్కరేసి బెల్లమేసి కాలు పోసి చక్కరేసి బెల్లమేసి
Yes, good evening.
Good evening, sir.
So today last class, right?
Yes, sir.
So what are the topic today? The topic is just wait. I will tell you. Principles and applications of paleontology, right?
Yes, sir.
So before that, already we discussed what is the paleontology?
Study of pollen grains.
Study of pollen grains. Already in the previous classes, we studied on how to development of pollen grains. You know, that is male gametophyte, right? Microsporogenesis. Microspores. So here, according to our syllabus, so principles and application of the paleontology. So the principles of paleontology. Here, so the study of external morphological features of pollen and spores is known as the paleontology, right? Yes.
Yes, sir.
The study of external morphological features of pollen and spores. So it is called as pollen paleontology. So the term paleontology was coined by Hyde and Williams in the 1945. 1945. So what is the meaning of pollen? It is the Latin word pollinate, which means it is a spread and distribute. Spread and distribute. After the development of the pollen grains, how it is spreading, how it is distribute. So that is the meaning of pollens, right? Just a minute. Okay, so the application of palynology, the study of palynology can either be basic or applied. So the applied aspect of palynology, it is includes geopalynology, like fossil pollen grains. So aeropalynology, pollen found in the atmospheric air. Some of the pollens, it is found in the atmospheric air. That is called as aeropalynology, right? Aeropalynology and latropaleontology. So medical aspects of the palynology like hay fever, asthma. It is due to this pollen grains only. So melitopaleontology. What is the melitopaleontology? Yes, melitopaleontology. Already we discussed in the previously. Melitopaleontology. Yes. ఏమైందమ్మా సుజాత?
Disease causing, sir. Palynology.
Melitopaleontology.
Telugu, sir.
Melitopaleontology. Melitopaleontology, it is nothing but study of pollen in honey. Honey. Got it?
Yes, sir.
Honey. తేనె ఐడియా ఉంది కదా మీకు, చెప్పాను కదా మొన్న. The pollens, the study of pollens in the honey, right? That is called as
Melitopaleontology.
సుజాత జస్ట్ వెయిట్ చేయండి అమ్మా, నేను ల్యాప్టాప్ లో లాగిన్ అవుతున్నాను. సుజాత
ఎస్ సార్, ఎస్.
ల్యాప్టాప్ లో లాగిన్ అవుతున్నాను, మొబైల్ లో షేర్ కావట్లేదు పీపీటీ. సో మళ్ళీ ల్యాప్టాప్ ఓపెన్ చేస్తున్నా, జస్ట్ వెయిట్. సో
Okay. How many types of Paley knowledges? So here, my voice is audible, Sujata?
Yes, sir.
Okay. So what is the geopaleontology? Yes, geopaleontology. The geopaleontology means fossil pollen grains, right? Fossil pollen grains. So aeropaleontology, pollen found in the atmospheric air, right? So another lateropaleontology. Lateropaleontology means medical aspects of paleontology like hay fever, asthma. So melitopaleontology, study of pollen in honey. And another one is forensic paleontology, the use of pollen grains in the crime detection. Okay, forensic paleontology means the pollen grains in crime detection. Okay, do you understand? Just please reply.
Yes,
Do you
sir.
understand?
Yes, sir.
These type of the paleontology are available. So here how to development of pollen. Already we discussed in the previous class, like the development of pollen grain starts in the sporanginous tissue of the anthers. The sporanginous tissue develops into the microspore mother cell, which undergo mitotic divisions, it resulting in the formation of 4 daughter cells arranged in the tetrahedral. It is a decussate, T-shaped, linear, isobilateral pattern. Already we discussed in the previously, right? Development of gametophyte, male gametophyte. Okay. How to development of pollen grains. Already we studied, right? Do you remember?
Yes, sir.
So coming to pollen dispersal. Pollen dispersal. So here, the dispersal of pollen is affected through different agencies like wind, water, and animals. So already we discussed this also. How to pollen grains dispersal, like some of the agents through this, like that is wind, water, and animals. It is the animals, it means mainly insects, right? So here, the flower pollen grains is not exposed to, and its fertilization occurs inside of the closed flower, but the pollen grains which are liberated into the atmospheric float in the air currents, and a few of which reach the stigma, produce a germinal tube to transport the male nuclei into the embryo sac, which results in the fertilization. Some of the atmospheric grains find their way to the nostrils of human beings while breathing and become a causative agent allergies like the asthma and hay fever. Thus, a large bulk of these grains produced become as a loss. These grains settle on the surface of the lakes, pit dogs also. It became embedded in the stratum and get preserved in the microfossils. These are the pollen dispersal. So next, chemical composition of the pollen and spores. What is chemical composition? The outer walls of the pollen and spores are composed in the pectineous substances called pollinin. What we call pollinin. So the chemical formula is C90H129OH5. In the spore of Onoclea sensibilis, there are traces of silica material. The protoplasm of the pollen grains contain proteins, amino acids, carbohydrates, lipids, vitamins, particularly the B group of hormones and enzymes, besides the traces of the inorganic substances such as potassium, magnesium, calcium, copper, iron, silicon, phosphorus, sulfur, and chloride. So, and high content of the pantothenic acid, adenosine are present in the pollen of Giames, and riboflavin in the pollen of the Almus glutinosa, and Almus inicana, and the rutin and quercetin are present in the pollen of the forestrin. These are the chemical composition. It is consists of the different pollen grains in the different plant species of the pollen grains. So here, coming to pollen structure and pollen and spore morphology. So coming to here, these the morphological characters of the pollen grains are an important criteria in the taxonomic consideration and interrelationship of plants at various taxonomic levels. So being microscopical structures, the history of pollen and spore morphology is closed. It linked with the invention and advancement of the microspore. So the pteridophytic spores have been examined as early the 1667 after Hooke invented the microspore. Later, the observation of pollen were made by the Greve, who recognized the pollen grains have different sizes and different shapes. The scientist Malfigi noted the presence of the germinal furrows in the pollen lilies. The lineage 1753 has taken the spore morphology into account of the species plantarum. Who is the written by species plantarum? The Linnaeus. The consequent of the rapid advances in the field of microscopy and introduction of phase contrast, interference, ultraviolet, X-ray diffraction, and electronic microscopy. It is SEM and TEM. So the morphological structure of exine was known as the great details. So here the units of pollen units. The pollen grains are produced within the anther of the flower. The pollen mother cells originate from the sporongenate tissue of the anther, which later divide mitotically, form a pollen grains. It is called as the tetrad condition. Already we discussed. So the pollen grains do not remain united at maturity and are disassociated into single pollen grain called monad. Single pollen grain, what we called monad. The condition is 4 is tetrad. Sometimes rare types like dyads is 2 pollen grains. Actans 8 pollen grains, and polyads many pollen grains also observed. So what are the morphological characters of the pollen grains? These are the different groups of the pollen grains. The first is the pollen units, polarity, symmetry, shape, size, apertures. So here, what are the in different groups? What are the first one is the pollen units. Polarity, symmetry, shape, size, aperture. These are the morphological characters and their terminology. So these are the different types of the pollen units. First one is the A, monad. This is everyone. Hello, visible my slide? Yes, please.
Yes, sir.
So just you observe the diagram. This is the round one. First A1 is the monad. Why we called in monad condition? Just tell me.
Single,
Yes.
only one is there, sir.
Yes, only it is represent only one pollen grain. So what we called? Yes.
Monad.
Monad. It is monad condition. Monad means mono, means only single, right? So the first A is the monad, and next B. What we called B?
Dyad.
How many?
2, sir.
2 is there. So dyads. Next C. What is the condition? How many pollen grains are there?
4, sir. Tetrad.
1, 2, 3, 4. Next D is tetra. Tetra means
4, sir.
tetragonal. So it is the number is 4, but the shape is different. Tetrahedral and tetragonal. Okay.
Yes, sir.
So here E, it is rhomboidal tetrad, because here the cells is 4, but the shape is different. What is the shape of here? E, rhomboidal shape, right? Do you understand?
Yes, sir.
Okay. So here C also tetrad condition, but shape tetrahedral. And so next, F is decussate tetrad. F, decussate. And G is the T-shaped. How it is look like? Already we discussed in the previous classes. So the shape of the pollen grains. So it is G is the tetrad shaped and T-shaped and tetrad. The shape is look like T, but here how many pollen grains? Only 4. So next H is the linear tetrad. How many? 1, 2, 3, 4. How it is arrangement? Linear. So I is the cryptotetrad. Cryptotetrad. Just to observe, tetrad means here also 4 pollen grains are there, but the shape is different. So J is the polyads. So polyads. Poly means yes, poly. Yes, J. You observe the J. This one. Yes.
Many, sir.
Many, right? Many. So here, what about K?
Pollinia.
Pollinia. So what is the meaning of pollinia? Already we discussed this pollinia we will observe in the which plant? Calotropis plant. Right?
Sir.
Calotropis. So all the pollen grains come together. It is a group of pollen grains. It is look like single one. A group of pollen grains. So that is called as pollinia. Right? So next step is polarity. The orientation of polarity is an important criterion in the identification and description of pollen grains as aperture position of primary phylogenetic and functional significance. So all pollen grains are in tetrad stage during the development, and the polarity is determined at this stage prior to their separation. So here the polarity, what is the meaning of polarity? The polarity expressed in the terms of the arrangement of the pollen grains in the tetrad. Yes, the polarity is expressed in terms of the arrangement of the pollen grains in the tetrad condition. So the end of the grains directed towards the center of the tetrad is the proximal pole, and towards the outside of the distal pole. So these are the regarding about polarity. Next, the part of the pollen grains which is nearest to the center of the tetrad is proximal pole, and towards the opposite side of the distal pole. So the majority, the imaginary line between the proximal and distal pole of the grain called is polar axis, which passes through center of the spore to the center of the tetrad. So the plane perpendicular to the polar axis through the middle of the grain is the equatorial plane, the equatorial diameter. So here, just you observe this diagram, the polarity, the A diagram is showing the tetrad stage, the B showing the length of polar axis and breadth of equatorial diameter in the monad grains. So these are the proximal pole, distal pole. In the upper side is the distal pole, the lower side is the proximal pole. So these are the regarding about equatorial axis. So next, so same thing. This is also regarding the heteropolar, praxipolar, isopolar, distal polar. So the pollen grains may be either a polar or polar. In a polar spores, poles or polar regions cannot be distinguished in individual spore after separation from tetrad. Among the polar types of the pollen grains are either isopolar or heteropolar, depending upon the demarcation of the between two equal and unequal polar bases respectively. The isopolar grains is distal and proximal places above and below in the equatorial plane, like this one. So the heteropolar grains of two faces are distinctly different, either in a shape, ornamentation, and aperture system. Thus, one face may have been opening and the other not. So these are the polarity. So heteropolar, isopolar. The pollen grains showing a slight difference between the distal and proximal faces are also called as perisopolar and subpolar. So for example, one face distal is convex and other face proximal is plane for the conveyance or. So next important one is the symmetry. So pollen grains or spores are symmetric or asymmetric. The symmetric grains are either non-fixiform without a fixed shape or fixiform with fixed shape. So the symmetry, it is the two types, symmetric and asymmetric. Asymmetric grains are either non-fixiform without any fixed shape and fixiform with fixed the shape. The asymmetrical grains have no plane of symmetry. There are rare in the occurrence. So the symmetric grains are either isometric, radially symmetrical. Radial symmetric grain, the shape is such as that any plane, including the polar axis, that passes through the will produce identical halveels. So these are the symmetry, how it is A to B, it is the radially symmetric, and C and D bilateral symmetry. So here, next, shape of the pollen grains. The shape of the pollen grains varies from the species to species. Shape of the grains are found to be useful in the spore or pollen identification. However, the shape may vary considerably within one grain type or even within the one species. The shape of the pollen spores may be circular, elliptical, triangular, rectangular, quadrangular, in other geometrical shapes. What are the shapes of the pollen grains or pollen spores? It is the circular or elliptical, triangular, rectangular, quadrangular, other geometrical shapes. So these are the polar view. Just wait, I will. So this slide is visible. So this type of pollen grains is available, discovered the scientist. So this type of pollen grains. Slide is visible? Yes, yes or no?
Sir.
So these are the non-angular, circular one, first one. The second one is the elliptical. B, angular. It is the triangular. So here two types, acute, obtuse. These are the. So next, quadrangular. These are the two types: acute, obtuse. This one the quadrangular. So here twin quadrangular. These are the acute and obtuse. So here these are the non-angular. These are the right side is the angular. In angular, it is the first one is the rectangular. These are the rectangular. So rhomboid. These are the rhomboid shape. So non-angular, it is the circular, elliptic. These are the elliptical and emarginate type and truncate. So here the Erdmann 1952 categorized the 8 shape classes based on the ratio of the polar axis and equatorial diameter. In the equatorial view, the ratio between the polar axis and equatorial diameter multiplied by the 100, it gives the indication of the shape. What is the principle? The equatorial view, the ratio between the polar axis and equatorial diameter multiplied by the 100. It gives the indication of the shape. So the various polar axis are equatorial diameter ratios are divided into different shape classes. Example is prolet, spheroidal, subprolet, parolet, oblate, and spheroidal, suboblate, paroblate. So these are the shape and classes. Paroblate, it is multiplied by the 100, it gives the shape. Oblate 50 to 75, suboblate. These are the Poland shape classes after the scientist Erdmann in the 1952. These are the shape classes. Paroblate and update, subupdate, update spherical, spherical, prolate spherical, subprolate, prolate, and perprolate. These are the different shape classes. Next, size. The pollen grains show a great variety of in this their sizes. The smallest pollen grains about 5 into 2.4 micrometer. It is the species is Myosotis palstris, and some members of the Boraginaceae. So here, Boraginaceae is the member of one family. So it is largest pollen grains below 200 micrometer in the diameter are observed in the Cucurbidaceae and Nyctagnaceae family members. So according to Erdmann in the 1945, categorized the different pollen size classes based on the size expressed length of the longest axis. So these are the pollen size classes after Erdmann. It is very small grains. It is less than 10 micrometers. Small grains, minute is 10 to 25 micrometers. Medium sized grains, 25 to 50 micrometer. Large grains, 50 to 100 micrometers. Very large grains, it is 100 to 200 micrometers. Gigantic grains, less than 200 size. So next, apertures. The morphologically apertures is an opening or thinning of the exine where the intine is usually thick. Physiologically, it is a germination zone. It is a germination zone. With regards to their position, apertures are polar, global or equatorial. The polar apertures are either monopolar, either in the proximal and distal pole, or bipolar, both in the proximal and distal phase. The global apertures uniformly distributed over the pollen and spore surface. Some tags have trema, a Greek word, means apertures. Pollen spores, they seem to have no special apertures, are termed as the in aperture and non-aperture. So here, these are the regarding of the pollen morphology. So the pollen morphology, it consists 6 types of pollen units: polarity, symmetry, shape, size, and apertures. Right? So next, coming to exine scarification. Exine scarification. The wall enclosing of the protoplasm of the grains consist of the intine and exine, right? Intine and exine. So the intine is haline. The exine consists of 2 layers: an inner homogeneous layer and an outer heterogeneous layer is the actine. So the actine is composed of the radial rows, columella, which are either free, their tips are united to form a layer called the tegillum. So the exine ornamentation. Exine ornamentation. The exine surface presents the various ornamentation patterns. The columella forming the actine aerosymptoms, it is free in the distal ends and the produce the pelate pattern in which the distal surface columella are bright and inverting the areas are dark. So and columella are arranged in the regular order and are fused to produce the reticulate pattern. Basing on the existence of the tigellum or other exine patterns like the granulose, spandulose are formed. So coming to one important aspect of these pollen grains, so we will go for the NPC system. NPC classification of pollens. So the NPC refers to, what is the meaning of NPC? Any idea? NPC refers to N means number, P means position, and C means character of the aperture in the pollen grains. What is the meaning of NPC?
Number, position, and character of the
Number, position,
and
character. So NPC is an artificial system of classification of the pollen or spore based on the 3 features of apertures, position and character. Number, position, and character. So the scientists Edman and Straka 1961 proposed in the NPC classification. Who proposed in the NPC system? The scientist is Edman and Straka. So according to NPC system, each pollen has arithmetic number consists of the 3 digits. The first digit reveals absence or presence of the aperture of, if presence, the total number of apertures present in the pollen grain, right? The second digit reveals the position of aperture, like digital or proximal, equatorial or latitudinal. The third digit is explain the character of the aperture. So it is circular or circular, oval or elongated, smile or compound. These are the regarding about the 3 digits Okay, the NPC system, each pollen as an arithmetic number consists of 3 digits. First digit, what is the reveals of first digits? Absence or presence of the aperture if present in the total number of the apertures present in the pollen grain. And the second digit reveals the position of the aperture, digital, proximal, equatorial, and latitudinal. So third digit explains the character of the apertures. So circular, oval, or elongated, smile, or compound. So these are the number is equal to n is equal to number, p is equal to position, c is equal to character of aperture composed of the NPC. So here the pollen grains without apertures called atrium and represented number of based on the number apertures pollen grains. So the classified monotrim, it is called as N1, ditrim N2, tritrim N3, tetratrim N4, pentatrim N5, hexatrim N6. Just wait, I will show the diagram. So here the classification of aperture based on the number. The N denotes the number of apertures present in the pollen grains. The pollen having apertures are divided into 7 groups. They are mentioned N2, N1 to N7. So in each group have the characteristic number of aperture. Example, N1 is the 1 aperture, N2 has the 2 aperture, and N7 has a 7 or more apertures. So N1 to N7 are referred as a monotrim, diatrim, tritrim, tetratrim, pentatrim, hexatrim, polytrim. Poly means above 7. The trema is a Greek means hole or opening. There are pollen grains where apertures are absent. Such pollen grains are called inaperture or atrium. So next, these are the N are placed under the number group. Atrium, there is no number. It is 1, N1, N2, N3, N4, N5, N6, N7. Anemotrim, it is above 7. So another special group, N8, is termed as the anemotrim, is treated where pollen grains have one or several irregularly spaced apertures. It is called as the under N8. So the classification of aperture based on the position. The before that based on the number. So here position P. So the P denotes position of aperture of the pollen grain. There are these 7 groups P0 to P6. The pollen grains with P0 group have uncertain or unknown position of the aperture. P1 group has the catatrim, and catatrim pollen grain have 1 aperture occurs the promaximal parts of the grain. P2 are the anectatrim, and up to it is P2. This group have 2 apertures, one of the proximal and other at the distal pole or distal end. P3 are called anatrim. It one at upper. This group have 4 aperture at the distal position. So P4 are the zonotrim. Zono refer to equatorial or sub-equatorial region. P5 digonotrim, where apertures are arranged in 2 or more zones parallel to the equator. P6 are panotrim, planto, all are wholly. The apertures are scattered all over surfaces uniformly. So here these are the position. These are the schematic representative of the position apertures in the pollen in the NPC system. So next one is the character of aperture of the pollen grains. Classification and basic of the character. The character groups of pollens are 7, and they mention C0 to C6. C0 group of aperture which character can't be established within the certain unit. The C1 groups of pollen have leptoma, thin place. Leptoma is a thin area aperture like function as a aperture pollen grains having 1 leptoma are called monoleft. The leptoma may be proximal or distal, and according to the pollen are proximal, cataleph, distal, analeph. CT groups are the trichotomoplate. Here the tricha is the three parts, toma means is the cut, colpate means is the depression of the furrow. It is the three branched apertures. The membrane and branches are more than times the longer than the breadth. The C3 cut aperture, C3 colpate type aperture, C4 like porate type, like a pore type, C5 like hole porate, C6 is the pororate. These are the classification on the basis of the character. So these are the schematic represent of the character of the some apertures present in the pollen grains and is number of NPC. So just observe it. These are the trim, left, trichotomocolpate, colpate, porate, pororate, coloporate. So next one is in the NPC system, grain is mentioned in the 3-digit number. In example, 343 inserted in the N3, P4, P3. So pollen grain having 343, N3, P4, 33. Titrine, jonopolate, N5, P5, C5 penetrate the digonocolpate. So the merits of the NPC classification and demerits, advantage and disadvantage. So advantage, the simple state of classification illustrate have a aperture of the pollen grain. It makes precise the description with the help of the NPC. The Pteridophyta, monocotyledon, dicotyledon can be differentiated based on this NPC system. The identification of pollen grains and spores is the essential prerequisite of applied the aspects of the palynology. Demerits, it's an artificial system of classification. Its synonym colpate and paracolpate do not fit the NPC system. Example, Tectona and Argimonis. So next one is here this is the like NPC system. So next we will go for the palynology and taxonomy, right? So the pollen morphology offer reliable data and evidence of taxonomic relationship. The palynological evidence may be used in place of the tags of uncertain affinities to suggest rearrangements and withdrawals and separations, as well as the forwarding other lines of the evidence. Sometimes the study of fine structural details in fossil pollen opens up the possibility of new sources of the phylogenetic evidence. So the major characters of the pollen grains that are used in the solving taxonomic and phylogenetic problems are the aperture patterns and exine stratification and ornamentation. So here the taxonomic and evolutionary importance of the pollen morphology may be at the specific generic higher levels. In many cases, the type of pollen of taxonomic characteristic and constant. Such taxon is called stenopollinous or unipollinous, and may be exclusive that group. Example, thick-walled pollen grains of the Gymnospermaceae, Gymnospermaceae of Australia. In other case, types of pollen may vary considerably in the size, aperture, stratification of the exine. Such tags are termed uripollinous or multipollinous. So stenopollinous tags are generally considered to be very natural. So Asclepiadaceae, Austiniaceae, Poaceae, Brassicaceae, and Lamiaceae are some of the stenopollinous families. So uripollinous condition seen in the Malvaceae is a primitive, while stenopollinous conditions seen in the Poaceae, it is advanced the characters. By using the NPC system, formulae can be derived and individual taxa and families. The families with more closely similar formulae must be considered more closely related than those the less similar formulae. So these are the regarding about the some useful aspects of the palynology. the relationship in the taxonomy. Next, coming to some brief introduction of aeropaleontology. The atmosphere abounds in the microorganisms and other organic matter, which includes pollen and spores. The study of pollen grains and spores in the air or it is called as the study of pollen grains and spores in the air. It is called as the aeropaleontology. The aeropaleontology is quite interesting because its application in the medicine, forestry, and paleobotany. So the airborne pollen grains are recognized as the principal case, like allergic. So sometimes we have suffering from the heavy fever and asthma, and because of these allergic types of the pollen grains, it is found in the atmosphere. Atmosphere means airs. That air we will take the air. So that time it is the pollen grains to affect our body. So due to this, we will face the asthma problem and heavy fever. These are the allergic pollen grains. So the information regarding the pollen content of the air is important and medicinal allergies. The pollen output is closely associated with the seed production of the forest trees. The paleobotany is interpretation of the fossil pollen data, knowledge atmospheric pollen content of the fossiliferous locality in the immense advance. The climatic factors such as temperature, humidity retard the dissemination of pollen in the air, but the pollen grains are resistant to high temperatures and are not destroyed in the air. So aeropaleontology is an useful field medicine, forestry, and paleobotany. So the pollen grains to great extent reflect the vegetation of an area and its surroundings. The pollen grain to great extent reflect the vegetation of area and its surroundings. The pollen and spores were preserved in the fossils, give a valuable record of the vegetational history of in the past. They are not destroyed in the medium which they are preserved because the exine of the grains is formed from the most extraordinary resistant material known to the organics, namely sporopollenin. So the resistance capacity of the protective walls can be evaluated from the fact of the pollen and spores were recovered even from the earliest geological formation. The data provided by pollen grains contain the fossiliferous rocks have been successfully made use of the tool in interpreting the fast vegetation of particularly of the quantity period in the different parts of the world. The composition of the pollen in the air of the pollen spectrum at the particular locality provides the from study of the present pollen and spores in the air, placed from the fossil pollen are recovered valuable clues could be had interpreted the first vegetation of the place. From the study of present pollen spores, the air it a place from the fossil pollen are recovered, the valuable clues could be had interpret the past vegetation of the place. These are the regarding about some brief introduction of the aeropaleontology. Next, geopaleontology. Like fossil paleontology, it is also called as the fossil paleontology. Plant fossils are representative of the fast vegetation. It is preserved in the geological formations covering the Paleozoic, Mesozoic, and Cenozoic areas. The fossil remains are only available the evidence of the historic evolution of the plant kingdom. So the fossil history plants evidence in the megafossils and microfossils. The microfossils, particularly the pollen and spores, provide ample evidence about the past vegetation. Apart from their purely botanical importance, these microfossils are used at a great advantage in the economic geology, such as in the correlation of the coal seams and oil fields. The tertiary and quaternary microfossils have their present in the equivalents in the identification of those pollens with present day genera or species is possible. The study of palynology is also help determining the age of rocks. The rocks are unknown geological age shall be dated in the microfossil evidence. The dating of marine transgressions such as the submergence and emergency of the coastlines has also been tackled by the pollen analysis. So next one is the melitopalenology. The plants offer pollen grains and also nectar to their insect the visitors. The honeybees collect the pollen and nectar for making a bee bread, which is an important item for the food of both adult bees and the brood. The pollen collected from the partial sources of vitamins, minerals, and amino acids in the honey. The study of relating to the various aspects of the palynology of honeys and related substance known as the melitopalenology. Nair, in the 1693, he made an extensive study of Indian honeys collected from the various places, such as analytical study has provided the useful data about the pollen and nectar yielding plants. The trained melitopalenologist can easily distinguish between the honey from the two different localities. He can also tell the time when particular honey was prepared. Honey said to be unifloral. It is dominated 50% or more by pollen of only one plant, and multifloral of it contains various types of the pollen considerably the percentages. Nair and Chaturvedi in the 1974 examined the microscopically 76 samples of in the Indian honey. They showed presence of the pollen belonging to the Eugenia, Sapindus, Putrangiva, Gythrus, Brassica, all nectar yielding plants. The Holo, Platella, Alinus, Borosus, and other palms pollen yielding plants. Such an analytical study provides the useful data about the pollen and nectar yielding plants of locality, which should be helpful the establishing the apiary gardens. So next, pollen and spore allergy. Some fungal spores and pollen grains initiate the allergic response to the susceptible individuals. The allergic people react to the potential allergens, being the allergic people react to the potential allergies, being the hypertensive to them, causing the several types of eye, skin, respiratory disorders. There are 4 types of major common respiratory allergic diseases, namely asthma, urticaria, eczema, and hay fever. The allergic diseases, namely asthma, and also the allergen is not pollen grain itself, but factors located with these factors induce the allergic diseases in the form of the hypertensive reaction. The allergens of pollen grains contain the protein or glycoprotein. Respiratory allergy is an acquired hypertensive reaction to specific components of the air spora. 2 types of allergy are recognized. So immediate allergy, it is after exposure, and other non-immediate prolonged exposure to the spores, mostly in the occupational environments. The allergens, the pollen grains, are located in the extracellular wall sites in the intestine and exine. This can be established using the fluorescent antibody methods. At growing number of population appear to the hypertensive pollen, spores, molds, and other microorganisms. By long-term monitoring of the occurrence of these allergens, their dispersal, interaction with the environment factors, annual and seasonal variations can be known to the sufferers and their doctors. With this knowledge, along with the pollen and spores factors of the allergic person, will be better position, avoid the unnecessary exposure to take right medicine in time and minimize the some of the daily troubles. So the plant known for the high pollen productivity are more significant for the pollen allergy problems. A survey conducted at the Bangalore has revealed that Casuarina equestifolia, which has been extensively planted under the afforestation programs, being the useful as a windbreaker, fire wound building material, is a high pollen producer. Allergic manifestations due to the pollen are high. So here in Hyderabad, GHMs also recently in the 2 years, 3 years back, they are started one plant. What is the plant name? Any idea? That is also, the plant is very dangerous to the respiratory, it causing respiratory disease. Any idea? GHMC. Yes. In the roadside plants.
No, sir.
This is very, very burning news in Hyderabad city, na? You find out. It is very dangerous.
Weed plant, sir. Weed
Yes.
plant. Weed plant.
No.
Weed,
Sorry?
weed, Parthenium, sir.
Not Parthenium. Just you find out. It is very, very important question also. GK question. GHMC, it is especially in the GHMC, they are planted in the very much of the, any roadside they are plants, but it is very dangerous to the birds. It is dangerous to the human beings also. It is causes the respiratory diseases. It is, they are released the allergic pollen grains. Just you find out the plant. Okay.
Okay, sir.
Okay. So and some medicinal properties of the pollen and spores also. It is some of the harmful pollen grains, some of the medicinal properties also there. So the medicinal properties of the pollen and spores, the Lycopodium powder consists of their spores are known commercially vegetable sulfur. This has been used medicinally for stomach disorders by the Arabian physician. The spores of clavatum are diuretic, dimplate, anti-spasmodic, and emmenagogue. The similar properties are known as the spores adiantum. The adiantum spores are recognized as a specific removed by the cough. The pollen of Cycas circinalis are narcotic, and those of typha have astringent properties. The pollen grains have also been observed in the process bacteriostatic activities on the intestinal flora of mice. There are several reports regarding the useful properties of the certain pollen tables, tonics, creams. The pollen tables are used in the treatment of the prostatitis. So these are the regarding about the medicinal properties of the pollen spores. So, and forensic palynology. Forensic palynology. So sometimes palynology also helps in crime detection, right? Sometimes palynology also helps in the crime detection. So like suppose in some incidents the crime is done. So the clues teams to identify the some like hairs and nails and something different. So spores also, the palynology also help in the crime detection. The several cases it has been possible to locate a site of the crime by studying cases. It has been possible to locate the site of the crime because suppose unknown crime is done. There is the crime, the pollen grains, where it is located, which plant. So it is help to the detect the crime also. So it mainly it has been possible to locate the site of the crime by studying of pollen in the samples collected from the dirt under the nails, from the mud stuck of the nails, and from the mud stuck to shoes and clothes of the victims. This is the use of the how to detect how to crime detection. So the samples collected from the The main purpose is it has been possible to locate the site of the crime by studying the pollen samples collected from the dirt under the nails, from mud stock to shoes, claws of the victims. The pollen samples from the stomach contents, it is excrements, have provided a clue several deaths in the Brazil. The deaths were ascribed to use of poisonous honey. The pollen are poisonous plants. Sergeneria lentesa, it is Sapindaceae family, were found in the stomach contents one of the victims who died after the taking of the poisonous honey. So this news is happening in the Brazil. So Brazil, so they are directly taken some honey that is poisonous, but we don't know this is poisonous or honey or natural. After he died, so so many tests done, there is no report. So the scientist go for the primary evidence is he's taken honey orally. So they are examined the honey samples. So the honey samples, they are found the poisonous pollen grains, right? So they are confirmed it is not a murder. He is not a suicide. He is naturally dead with the causes of the poisonous pollen grains, right? These are the regarding about the forensic palynology. Yes. So these are the principles and applications of the palynology. Okay. So here, how many types of palynology? Many times they are asking questions in the GK question, like in JL or Group 2. And so once again, I repeat. So geopalynology. What is the geopalynology?
Fossil pollen.
Yes, fossil pollen grains. Next, aeropalynology. Yes, tell me.
Air, sir.
Yes.
Air, environment.
Yes, the pollen found in the atmospheric air. Okay. Next, latropalynology. Yes, tell me. Latropalynology.
Asthma and fever, hay fever.
So that is medical aspects of the palynology. What is this? Medical aspects of the palynology. Next one is like what is the aspects of the medical palynology, like hay fever and asthma. So next, melitopalynology. What is the melitopalynology? Yes.
Honey, sir. With the pollen.
Yes. Once again, repeat, please. Melitopalynology. Yes, melitopalynology.
Honey,
Yes.
Honey, sir. Honey.
honey and related substance.
Yes, not related. This is fixed with only the study of pollens in the honey samples, right?
Honey samples. Okay.
Honey samples. It is very, very important. Please remember. Melitopalynology, the study of pollens in the honey, right? So next, forensic palynology. Yes, what is the forensic palynology?
Crime detection.
Yes, detection of the crime through the pollen grains. That is forensic palynology. So here in this chapter, you read the pollen morphology is the important question. Pollen morphology is the important question, and NPC classification is the classification also important, and they are asking compulsory short questions as a melitopalynology, or otherwise aeropalynology, otherwise latropalynology, otherwise geopalynology, otherwise forensic palynology. Also compulsory question, these types of question, one short question definitely. Just what is the related this, how to going on, just that matter is enough to in the short answer. In this, the NPC classification is the long question, and morphology of pollen grains also, it is long question. These two are the very, very important in this chapter. And the short answers is the melitopalynology, forensic palynology, aeropalynology, latropalynology, geopalynology, right? Yes?
Sir.
Okay. So today onwards, our syllabus successfully completed. Okay.
Yes, sir.
Okay. Thank you.
Okay, sir. Thank you, sir.
Thank you.